Hello friends, my name is Nikhil Shah. I am working as an assistant professor in the Hewadi College Hewadi at Department of Microbiology. Okay, so today we are seeing the concept of Operon. First, we have to see what is Operon. So the Operon, it is a functional unit of genetic material which is found into bacteria as well as the archaea. It consists of different kind of set of genes into which there is a repressor, operator, promoter as well as a regulatory genes is there. So the it consists the set of genes which related function along with the regulatory elements all transcribed together into a single mRNA molecule. That means there are some set of genes which are arranged into the operon into double stranded DNA, and it will be converted into single stranded mRNA, and after formation of mRNA there is a formation of bacterial important protein into the operon. The operon concept help bacteria efficiently organize and regulate their genetic material to suit their dynamic, often as well as a challenging environment. That means operon which help to bacteria to adjust into the environment. So operon provide a mechanism for bacteria to evolve and adapt rapidly, changing in their environment. Mutation in regulatory elements that changes the operon can lead to promote or can provide a new function into the bacterial cell. Operon consists of different genes which can perform different function like promoter region, operator region as well as a regulatory region. So into the diagram we can see that clearly. This is the bacterial cell. Into that bacterial cell there is a double stranded DNA which can contain the promoter region which can promote the function, operator region which can operate the function, and structural genes which can do the transcription and form mRNA. After the formation of mRNA there is a formation of bacterial important protein with the help of operon. So first we have to see the first region of the operator. This is called as promoter region. So the promoter region in an operon play a important role that regulating gene expression by facilitating the initiation of transcription. Initiation of transcription means that is the transcription. This is the point where transcription will be happen. Okay, the promoter region contains a specific sequence onto the DNA where RNA polymerase will be bind and initiate the transcription. Initiation means this is the point where transcription will be start. RNA polymerase is the enzyme responsible for synthesizing the RNA from the DNA template. DNA it is a double stranded and RNA it is a single stranded. How can this is possible? It is possible with the help of RNA polymerase. RNA polymerase it is an enzyme which breaks down the double stranded DNA into single stranded mRNA. That means this is the transcription. So promoter are the specific DNA sequences that are signaling starting point for the transcription. So when there is a promoter region into the bacterial cell, there are some sequence which can work as a signaling molecule for the RNA polymerase. The signaling molecule in the sense of when RNA polymerase come and it will bind onto the promoter region at specific sequence. This is the mechanism and this is the function of promoter region. So promoter often contains specific DNA sequences called as a consensus sequence. Consensus sequence means where RNA polymerase will be bind into the consensus sequence like Tata box. It is also a consensus sequence. So these sequences help ensure that accurate binding of RNA polymerase to start the transcription process. What is the function of consensus sequence? This is the working as a signaling molecule to start the transcription process. So the transcription begins with the binding of RNA polymerase to a specific region onto the DNA. It is called as a promoter. So what is promoter? Into the promoter there is a presence of consensus sequence where RNA polymerase come and bind. This is called as a promoter region. Now we are seeing the different region into the operon. It is called as a operator region. Operator means which maintain the operating system. The operator region, it is an operon or it is a segment of DNA that situated into the adjacent to the promoter. Into simple language, after the promoter there is a presence of operator region. It acts as a regulatory region. What is the function of operator region? So it works as a regulatory region. that controls the access of RNA polymerase to the structural genes within the operon. The operator region contains binding site for the regulatory protein. This is known as a repressor. Into the operator region, if suppose there is enough formation of bacterial protein, the process will be stopped. But when the process will be stopped, when there is a binding of repressor region into the operator region. In the presence of repressor protein, the operator acts as a switch that can block the path of RNA polymerase from transcribing the structural proteins or structural genes into the structural proteins. The repressor bind to the operator region. When the repressor will be bind to the operator region, this is the stoppage of gene expression. That means there is a stoppage of formation of protein. So into this diagram we can see clearly, if suppose there is a formation of operon into the bacterial DNA, there is a promoter region, operator region, and these are the structural genes which can form the structural protein. But how the structural protein will be formed? The RNA polymerase will be bind at a promoter region. It can transcribe the different kind of protein, and this will be enough for the bacteria. But if suppose there is enough protein formation, at that time this activity will be stopped. But when the activity will be stopped, there is a binding of repressor protein into the operator region. This can stop the activity. This is called as a operator region. Operator region work as an operating or maintaining site of the protein synthesis. So last we can see the structural genes. So the structural genes into the bacteria that are crucial or important into the various biological process that play a key role in overall functioning of the bacterial cell. A structural genes in an operon, it is responsible for encoding a functional product, typically a protein. After the formation of a protein, this is important for the bacterial cell. So structural gene encoded the information needed for the synthesis of specific protein. If suppose into the bacteria there is a formation of specific protein, at that time structural gene will be important for the synthesis of the, these are the important or the special kind of protein. So the protein are essential for the structural function and regulation of a cellular activities into the bacteria. What is the importance of protein into the bacteria? These are important for the maintaining the structure as well as doing the intracellular function as well as doing all kind of cellular activity into the bacterial cell. So many structural genes encoded for the enzyme that catalyze the biochemical reactions. There are some proteins which are important for carry out the biochemical reaction which are present into the bacterial cell. These enzymes are vital for metabolic pathways. What is the importance of these proteins? These proteins are important for doing the metabolic pathways into the bacterial body, nutrient utilization as well as the production of energy into the bacterial cell. So the products of the structural gene contribute into the various cellular function. What is the importance of synthesized protein? That means there is a formation of protein which are important to do the cellular function like cell division, cell wall formation, cell wall synthesis, DNA replication and other bacterial process which are important to stay or maintain the bacteria at a environmental condition. In pathogenic bacteria there is a formation of disease, but when the disease will be formed into the host, when there is a secretion of uh, pathogenic protein which are coming from the pathogenic bacteria, that means the structural genes may code for the virulence factor into the pathogenic bacteria. There are some protein which are important for to do the virulence factor creation. In that enable the bacteria that invade the host tissue into the, there are some pathogenic bacteria which can invade the host tissue, that means they can create the disease into the host. So these are the overall structure of operon. Now we can see in details also by structurally. So this is the double stranded DNA which contain, there are some promoter region, operator region and there are some structural genes. So promoter region which has consensus sequence where RNA polymerase come and bind and the transcription will be done, and after the binding of RNA polymerase there is a formation of mRNA. After the formation of mRNA there is a formation of structurally important protein which are maintain the bacterial health or which are useful for to adapt bacteria into the environmental condition. What is the operator region? Operator region, it is a stoppage pathway or it is a stoppage site where RNA polymerase will be stop their activity when the repressor will be bind. And what are the importance of structural genes? Structural genes which are important for synthesizing of the structural protein. So this is the overall concept of operon. Thank you.